All right, let's do another activity and another blast exercise. So the information we have is that the following DNA sequence was isolated from a bacterium that lives in the gut of an insect. So I'm copying the sequence and pasting it again into blast. And because we don't know, so rRNA and messenger RNA, you can't distinguish. If you're not told whether it's an mRNA or an rRNA, you have to be careful what database to choose. Okay, because we don't know whether it's a 16S or whether it is a messenger RNA. We're just going to use a standard database. Because if it's a messenger RNA for like a toxin or something and you're choosing rRNA, you won't get a hit. So that's really important. So not knowing what the sequence is, we use standard nuclear database because we know it is a nucleotide sequence, not a protein sequence. And then again, copy, choose your database, and then we blast. Let's see what we got. We blasted our sequence of the bacterium that lives in the gut of an insect. What could it be? All right, Borrelia burgdorferi. Well, for many of you, probably doesn't mean anything. So let's see what the paper was that submitted the information. All right. Definition reference. Here is a paper. Vector-borne disease section. All right. Doesn't still help us a lot, right? So we have to do some more research of what Borrelia burgdorferi is. But we can just use that information and go to our database. We go to PubMed. Let's see PubMed. And Borrelia burgdorferi, what does it say? Lyme disease. It is the causative agent of Lyme disease. All right. So we figured out, or we can figure out what the insect is in what gut it was found. It was a tick. All right, let's go back to our results. So Borrelia burgdorferi, different type of strains. The E value is really high. So again, it's very unlikely that it's just by pure chance. So there's a good chance that the sequence we had is actually from a Borrelia organism. The query covers 100%, so all of the nucleotides are used, and it is 100% alignment. So again, we are very, very sure from our analysis that the sequence we entered was part of the 16S of Borrelia burgdorferi, the organism that causes Lyme disease. All right, you can look at the alignment. It is perfect. It's almost a perfect match. And again, we can look at the taxonomy and understand Borrelia a little bit more. And there are lots of different Borrelia burgdorferi strains. So this is the main organism, different isolates, but they all cause Lyme disease and they're found in ticks. So again, detective, job well done.